Hi everyone, welcome to the Basics lecture on ATP, enzymes, and redox reactions. This is one of my favorite units because the parts of it fit together in a really interesting way. We'll be talking about the basics of energy in biology, including ATP, enzymes, and redox reactions, and then we'll talk about how energy is captured and released in live organisms. To start things off, let's think about cellular work. We've talked about how cells accomplish several types of work so far. ATP, for instance, is used to pump solutes across a membrane. In doing so, the ATP molecule is changed to an ADP molecule in a phosphate group. Similarly, ATP provides the energy that is used to move a motor protein along a vesicle. In this molecule, ATP is used to make the reactions in the cell proceed that are endergonic or require energy. All molecules contain a certain amount of free energy. Some reactions in the body have high energy reactants and low energy products. These reactions have a net release of energy and are thus called exergonic. The classic example of this is cellular respiration. Reactions that take energy from their surroundings have products with a higher free energy than the reactants. These reactions are called endergonic and produce a net storage of energy. The classic example of this, of course, is photosynthesis. Adenosine triphosphate is a molecule used for short-term energy work in the cell. It's a nucleotide, just like the ones we saw in the nucleic acid lecture, though it has 3 phosphates with its sugar and its base. The 3 phosphate groups are negatively charged and tend to repel each other. This instability makes it a molecule with a high free energy. When a phosphate is removed from a hydrolysis reaction, this is called the dephosphorylation of ATP, and the resulting molecule has a lower free energy. Graphically, you can think of the hydrolysis releasing that free energy that can then be used in other bonds. Now let's leave behind ATP for a minute and talk about free energy in a different context. Many reactions in the cell are exergonic, meaning that the end product is a lower free energy than the starting product. But there's always energy required to break the first bonds in the reaction in order to create the new bonds. This input of energy is called the activation energy of the reaction. And the energy is provided by the kinetic energy of the molecules bumping into each other. But in biological systems, this happens too rarely to make the reactions useful. So an enzyme is a molecule, usually a protein, that helps the reaction proceed. Let's look at how this happens in a cartoon form. Say that these red and green blocks are the reactants. We're going to break each of them apart and create 2 new products. This available enzyme is floating around, and these molecules are able to diffuse into its active site. They are the substrates for the enzyme, and their binding changes the shape of the enzyme. Enzymes are very specific and will only bind to one type of substrate. The enzyme-substrate complex brings the 2 reactants together in such a way that it allows the bonds to break more easily with less energy required. Once the reaction has occurred, these new products are shaped differently and are not attracted to the active site of the enzyme. The products diffuse away, and the enzyme is freed now to repeat the process with new substrates. Notice that the enzyme does not use ATP and is not used up in the reaction. The end result of enzyme activity is to decrease the activation energy of an exergonic reaction. This makes the reaction occur more often when the enzyme is present and rarely occur when the enzyme is absent. Almost all biological reactions utilize enzymes, usually named with an ACE on the end. Here, sucrase is the name of the enzyme that helps the hydrolysis of a disaccharide sucrose into 2 monosaccharides. Not only do enzymes make the reaction happen more easily, but they allow the cell to control when the reaction occurs by providing or taking away the enzyme. Our final topic is on oxidation and reduction. It turns out that the metabolic processes of cellular respiration and photosynthesis are based on oxidation and reduction reactions. Reduced molecules are defined here as molecules to which electrons have been added, along with hydrogen ions that tend to come with electrons. Sugar is a reduced molecule, as is water. You can recognize reduced molecules in a biological reaction because they have more hydrogens. Oxidized molecules have had electrons removed along with their hydrogens. Oxygen is an oxidized molecule, as is carbon dioxide. Notice that we sometimes use these terms, oxidation and reduction, as adjectives and also as verbs, so be careful when you use them in a sentence. A redox reaction is moving electrons from one reactant to another. Glucose is a reduced molecule, and the electrons here are high energy electrons on outer orbitals that are relatively easy to pop off. So I'll draw them here. This is what makes glucose a high energy molecule, is that the electrons are so unstable. Cellular respiration is the process of removing these electrons one by one and putting them and their hydrogens on oxygen. Water, the resulting product, is a very low energy reduced molecule where all the electrons are very close to the nucleus. Okay, so this reduction of free energy from electrons that are far away from the nucleus to putting them in a molecule where the electrons are close to the nucleus releases energy. This energy is captured in the form of ATP. You can summarize respiration then as turning a high energy reduced molecule into a low energy reduced molecule and capturing the change as ATP. Similarly, photosynthesis is the process of taking a low energy reduced molecule, water, and moving the electrons to a high energy reduced molecule, sugar, utilizing light energy in order to make this change. This completes our basics video for today. Write out the answers to the following questions to study for the basics quiz, and thanks for watching.